everyone. I'm here to explain to you today how to do day 1 of Lab 8. And so you probably have a copy of the lab handout. It is for size exclusion chromatography. And the objective of today's lab is to isolate using a size exclusion column 2 fluorescent proteins. One is called GFP for short, the other one is called BFP for short. And you know from the lab handout that GFP stands for green fluorescent protein and BFP stands for blue fluorescent protein. So the first part of this lab is to set up a Cefidex column and pack it with the Cefidex beads. Then we're going to wash the column with a little bit of elution buffer. After that, you're going to load the protein mixtures into the column and isolate elution fractions. You'll choose the fractions that have the most GFP and BFP in them. And then with the help of a test called the Bradford protein assay, you're going to help measure the relative amount concentration-wise of isolated GFP and BFP protein. All right, so let's get started. So to set up the column, you should have a ring stand in front of you, and the clamp on the ring stand is situated in such a way that it'll allow you to clamp the column into it and also fit an empty beaker underneath it. The beaker is going to catch all of the liquid that you are not meant to save, which is going to be waste, basically. All right, so this is the column itself. It's an empty tube. And the Cefidex beads are not in the tube right now. There's a little piece of foam that'll keep the beads in the column. There's also a small cap on the bottom that acts as an on and off switch for the flow of liquid going through the column. So once you take the cap off, you want to keep it because that's the only thing that will, when you put it back on, help you stop the flow through the column. There's another cap, it's a blue one at the top that you can take off once you've clamped this tube into the ring stand. You can take this blue cap off and leave it off for the entire lab procedure. All right, so to clamp the tube into the column or into the stand, you want to make sure that the thickest part of the tube is what you're clamping in, so you're not going to see that. And before you tighten it all the way, you want to do a couple of things. First is you want to raise the column up enough so that there will be some clearance between the bottom of it and the top of this beaker. That way you can take the little white cap off without a whole lot of trouble. Secondly, before you tighten things all the way, you want to make sure from all sides this tube looks completely straight. So even though it might look straight from one angle, you want to check it from all angles to make sure that it's completely vertical from all sides. And once you do that, you can pop this blue cap off and start applying the Cefidex beads into it. So the Cefidex is suspended, those would be these white beads, suspended in a clear elution buffer. And in order to make sure you're getting an even distribution of these beads before you apply them to the column, you want to invert the tube many times like this. Don't shake it so you don't create air bubbles, but you want to invert the tube several times so that the beads are not clumped at the bottom of the tube. And once you have the beads fully suspended in the buffer, you'll know when you tip the tube over and you don't have any beads clumped at the bottom, then you're ready to go. And you want to keep on doing this anytime you want to take any of the Cefidex slurry out. So to apply the slurry, you're going to grab a clean transfer pipette like one of these. You're going to suck up some of the recently resuspended Cefidex beads. And then you want to apply the beads to the inside of the column gently by touching the tip of the transfer pipette to the inside of the tube. And then slowly, in a smooth motion, press down on the bulb of the transfer pipette. That way the liquid is streaming down the side of the column and not dripping down the middle of it. And you just keep on repeating this until you get the Cefidex suspension filled all the way up so that the narrow portion of the column is completely filled with it. Once you've done that, you want to take this little cap off of the bottom of the tube and let some of the liquid drip out of it. And if you do that, you will see some of the beads will start to compress even further. That's what we want. That's the packing that we are aiming for. all right? And as long as liquid is dripping out of the column, you don't have to put that little cap back on. So as the beads compress, you will have to add more of the slurry into it so that you get the level of the slurry back up to where we had it originally. And just keep on repeating that until the column is completely packed and uniformly compressed with those white beads. Okay, so now that you have the column fully packed with the Cefidex beads, we're going to have to wash the beads very quickly using a little bit of elution buffer. This basically just flushes out the pores that you know are built into the beads. That way they can reach their full potential of separating proteins by size. So to apply the elution buffer, you'll need another fresh transfer pipette. And the lab procedures say to add roughly 1.5 pipettefuls of this elution buffer. So that's a very rough measurement. And what we basically mean by 1.5 is you want to suck up roughly a whole length of this pipette as one, and then another half of its length as a half. And to apply it, you want to do the same thing you did to apply the beads in the first place. Touch the transfer pipette to the inside of the column, and you want to release the liquid in as gentle of a stream as you can so it runs down the inside surface of the column. And the column should fit all 1.5 pipettefuls of the elution buffer, so you can add it all at the same time. You just want to add it as slowly as you can. Once you've done that, what you want to do next is wait for all the elution buffer to drip out of the other end of the column so that you don't see any elution buffer left on the top of the beads, and then you'll cap it to stop the flow before you move on to the next. All of the elution buffer wash has dripped completely through the column. You'll notice at the top of the column you don't see any clear liquid anymore. That's when you know to stop the flow. So I put the little cap back on the bottom of the tube. And next, what we're ready to do is actually use the column for the size exclusion. So you're going to use it twice. The first round is to isolate GFP from a larger protein mixture. Second time, you're just repeating the same steps to isolate BFP from a second larger protein mixture. So here's how you load the protein mixture. First one containing GFP in it to the top of the column. And you can see in the protein mixture that the GFP is glowing just from the light that's in this room, all right? But if you recall, it isn't just GFP that's in there. So I'm gonna load 200 microliters of this mixture to the top of the column. And again, you want it to run down the sides of the tube in as gentle of a stream as you can. So 200 microliters, I'm gonna suck into this tip. And if you're a little bit short, that's okay, just go with what you have. And I'm gonna apply this tip to the top of the column, touching the inside, and slowly dispense this protein mixture to the top of the column. And you can see that it's on top of the Cefidex beads as we speak, all right? Once I get rid of the tip, now you're ready to let the protein mixture run through the column. And before you do that, you don't want to catch the liquid coming out as waste anymore. You want to replace this waste beaker with one of the fraction tubes. These are little microfuge tubes that I labeled as G1 through G4, all right? One of these microfuge tubes, the first one, labeled as G1, I'm going to open it and make sure I center it underneath the column. And if you need to, feel free to lower the column a little bit closer to the tube. But still maintaining a little bit of space so you can take that little white cap off of the bottom. All right, and now you're ready to let things drip. And things will drip so slowly that if for some reason you're a little bit off center and you lose a drop, it's really not a big deal. And there's so little liquid on top of the column that it'll stop dripping eventually. That's when you know to cap the tube again before you move on to the next. So now that the protein mixture containing GFP has fully entered the Cefidex beads, and you can see that because the GFP in the mixture is visible and it's sitting only at the top of the column. Now you're ready to push the rest of the mixture through the rest of the beads in the column by adding more of this elution buffer. So with a clean transfer pipette, you're going to suck up any amount of the elution buffer, and you're going to apply it very gently to the top of the column like you did with the wash step. The key here is to keep on applying buffer at all times to get it all the way to the very top of the tube, because the more liquid you maintain on top, the more constant and fast the flow rate will be for you. And again, by letting the liquid slide down the inside of the tube, it'll be a little bit gentler of an application than letting it drip down the middle. All right. So we're just going to pretend that I've added elution buffer all the way to the top of this tube for the sake of time. And once you've done that, now you're ready to release the little cap again, and that'll restart the liquid flowing through the column. And you will keep on collecting liquid into this first tube that I've labeled as G1 up to the mark, that little Sharpie mark, which is roughly 1 milliliter. And what you'll notice is, as there's liquid on top and it's moving through the column, this band of GFP will also migrate down and drip out of the bottom of the tube. So you want to maintain the maximum amount of elution buffer on top of the column at all times. And once you've filled up this fraction tube to its Sharpie line, you can just slide the tubes over in the rack to G2 and keep on filling it until you get all 4 of these fractions filled. Once you've isolated all of your GFP and BFP fractions, you'll want to check your fractions using a UV light. To choose the fraction, it should be really 1 out of the 4 that has the most GFP or BFP in it. And what you're looking for is the fraction with the greatest amount of fluorescence. So here's some examples. Here's an example on the left of a fraction that has no GFP or BFP in it. And then this is a fraction that I isolated with a large amount of GFP. So you can see it glowing green. This one on the right is a fraction that has the highest concentration of BFP in it. And it's really the BFP that you need the UV light the most, because it won't fluoresce all on its own with the light in your room. So once you've chosen the 2 fractions, one with the most GFP in it, and the second one with the greatest amount of BFP in it, you're ready to move on and do what we call the Bradford protein assay. Now, the assay is basically a color test. So what you're generating to measure the concentration of protein that you've isolated is the presence of a blue dye. And that blue dye is created when a chemical called Bradford reagent, which is in this foil bottle, is added to your protein sample. And generally, the more protein you have in a sample, when it comes in contact with the Bradford reagent, the bluer, the more of that blue dye will be created. So before we can do the assay, we have to calibrate the spectrometer, which is going to take absorbance readings of that blue dye before we can use the machine. So in order to calibrate the spectrometer, you should have in your lab procedures a set of instructions that you're going to follow to the letter. And in order to do the calibration, you need to stick into your spectrometer eventually a blank. And this blank is going to consist of the Bradford reagent all by itself, just to tell the spectrometer not to measure the absorbance of the reagent if there is any left inside of the cuvette. All right, so we're going to apply the Bradford reagent to cuvettes. You've used cuvettes before in previous labs. They're plastic and they're disposable. And it's generally 1 cuvette per sample because they're disposable. And into this cuvette, which is going to act as my blank, I've put in 1.5 milliliters of Bradford reagent. So notice it has a brown color. All right, and I'm going to stick this cuvette into the spectrometer. There is a little arrow on one side of the cuvette that you want to make sure you're matching to the same looking arrow in the sample holder of the spectrometer. That way you know that the cuvette is oriented properly, and then you're going to stick it in. What you'll be reading is absorbances on the LabQuest display, which looks like this one. Before you calibrate this display, there should be no number inside of the red box. And after you calibrate it, there should be a number there. And with the blank in there after calibration, it should have an absorbance of 0.000 because that's the entire intention of the calibration. All right. So to calibrate the spectrometer, again, you're going to follow these procedures in your lab handout. It'll tell you using your stylus or the eraser or pencil what to tap in the LabQuest display, and pretending that we tapped all of those correct things with my blank in there, there should be little to no absorbance. Sometimes the number will wobble a little bit, but it should be virtually zero. In order to do the Bradford assay, one of the first things you need to do after you've calibrated your spectrometer is to boil a little bit of your two chosen fractions. So I've transferred 100 microliters of my chosen fractions, both the GFP and the BFP one, into separate microfuge tubes. And then I want to boil them in a boiling water bath. So in order to start the boiling water, you want to turn on your stir hot plate. And on this stir hot plate, there's a temperature dial. I'm going to crank it all the way up. All right. What you're looking for is a nice roiling boil. And once you get that, you're ready to boil your two 100 microliter samples of your chosen fractions. To get these samples ready for boiling and to ensure that the tubes they're sitting in don't explode while they're boiling, we have some reusable microfuge tube cap locks that look like this, and they slide over the top of the cap of a microfuge tube. That's what you want to do to them to keep the cap closed while they're boiling. And once you do that, you're ready to stick them into a floaty that looks like this one, and that'll keep it from submerging in the boiling water. And then once this reaches a full boil, you'll stick it into the boiling water and boil them. All right, so remember that you're only boiling 100 microliters of each of your chosen fractions. The remainder of your fractions you want to not boil and save what's left for the next lab, which is lab 8, day 2. To the 100 microliters that you boil, once you take them out of the boiling water bath, you want to let them cool to room temperature roughly before you're ready to move on to the next step with them. While you're waiting for your samples to cool, one thing you can do is prepare a standard, which would be 7 parallel dilutions of a protein called bovine serum albumin. That's going to help you draw a standard curve as part of your post-lab. So here's a stock solution of bovine serum albumin. The bovine serum albumin, or BSA, has already been diluted to a concentration of 2 milligrams per milliliter. So what you're trying to do is you're trying to make parallel dilution 7 of them according to the lab procedures of this stock solution. So what you should have in your lab procedures is a table that looks like this one. Using the parallel dilution formula, you're supposed to calculate V1 for each of these 7 parallel dilutions. That's the volume that goes into the first column. Each parallel dilution should add up to 100 microliters total. So once you've calculated V1 and entered it into the first column, subtracted from 100 microliters, that'll get you the amount of distilled water that's needed to fully prepare each of these parallel dilutions. And these will be your recipes to make all 7 of them. So following this recipe in this table, I prepared all 7 of these BSA parallel dilutions, and I have them here in these 7 microfuge tubes. I put the water in them first, and then I followed that up by adding the BSA stock solution to each one of them, one at a time, changing my tips every step along the way. Once you do that, you want to give each of these dilutions a quick tap to mix the contents. And if things are splashing around, you can always spin them very quickly in a microcentrifuge just to collect everything to the bottom. So once you've done that, you're ready to move on. So I have transferred all of my BSA dilutions, 7 of them, into their respective cuvettes. I've also transferred the right amount of my boiled GFP and BFP chosen fractions to their respective cuvettes. So now I'm ready to actually start the Bradford assay by adding 1.5 milliliters of Bradford reagent to each of the cuvettes, and I'm going to add them in the order that I've situated them in my rack and start timing 5 minutes with the first one. All right, so what you will notice is, and we're just going to pretend that I've added it to all of my cuvettes in the order that I've situated them. What you'll notice is the more protein is in that particular sample, when it comes in contact with the Bradford reagent that I've just added, the bluer it's going to turn. So this is an indicator of the concentration of protein that's inside of that cuvette right now. Once the 5 minutes are up, I'll stop the timer. And in the same order I added Bradford reagent to these cuvettes, I'm going to stick them one by one into the spectrometer and measure their absorbances. So the absorbances should show up on the LabQuest display. Those numbers I will record into the table that's in the lab procedures. It looks like this. So I have spaces for all 7 BSA dilutions. I also have blanks for the absorbances of the 2 boiled GFP and BFP fractions. And it's this data for the 7 BSA dilutions that you will use to create a standard curve that's part of your post-lab. Once you're done taking all of the absorbances, the only thing that's left is to make sure that you save the remainder of your chosen GFP and BFP fractions. That's what you're going to give to your instructor to save for the next day's lab. And then clean up the lab space.